crucial to implementing. It's very powerful. It's very simple. Like I said, we're very excited about it. And Connor has lots of ideas how he can use it for your research. So Connor came to our facility almost 2 years ago now. He was at the USDA since 2016. Before that, he got his PhD at UT Southwestern. He's an expert in microscopy. He's also really gifted with respect to electronics. And so there's a lot of things that you can talk to Connor about, get advice on with respect to imaging, image analysis, in addition to this high-tech thing, CODEX he's going to tell you about today. So I'll get out of the way with that. So go ahead, Connor.
Thank you very much, Ken. And so today we'll be talking about the CODEX multiplexing platform. It's available in the core facility. This is just a brief snapshot of the kind of data that you can acquire from the CODEX system. This is 7 of the 14 probes that were actually used in this system. This is mouse pancreas tissue. This happens to be an islet. And what I hope for you to appreciate here is that we're actually able to get quite a high density of data out of a single CODEX run. For any of you who are not aware of what our website is, I just want you to be aware that we have a website with all the information regarding all the systems in our facility. And you're welcome to look up more information here or send me an email if you're interested in any of the things that we talk about today. So just I wanted to go over a brief overview of the things that we have, the equipment that we have in the facility. We have several confocal microscopes, the first of which is the Olympus FV1000 confocal microscope. It's equipped with a deep sea multiphoton laser. We actually have 2 Leica systems, both of which are SP8s, one of which is an upright confocal system with a single multiphoton laser attached. And then we have an inverted SP8 system with dive detectors, very sensitive detectors. And it also has an attached FLIM system, a fluorescence lifetime imaging system. And then soon we'll actually have a digital light sheet attachment as a demonstration unit in the facility. And if you're interested in preparing samples or using the light sheet that will be in the facility for a short while, please let us know and we'll be happy to arrange a consultation with the Leica rep to get your samples prepped and arrange some time for you to get onto the microscope for the demonstration. We've also recently upgraded our Nikon Yokogawa spinning disc system. This is a confocal system that is well suited for live cell imaging. If you have any interest in doing some live cell imaging, please let me know. And then, of course, today we'll be discussing the Kient's automated microscope system in conjunction with the Akoya Biosciences CODEX multiplexing platform. A little bit more detail about that later in the talk. And then, of course, after generating copious amounts of data on our microscope systems, you'll need a way to analyze them. We have 4 image processing workstations in the facility with a high degree of RAM and visual processing capabilities, as well as some high-end imaging software available, including ImageJ and its components Fiji, Metamorph, Halo, which is a software program that will be used in conjunction with the CODEX system, and is something that I'll be demonstrating at the end of the presentation today. Flow Render software, MRS 9.7, Olympus FlowView, which is in conjunction with the Olympus microscope, Leica LASX, which is in conjunction with the Leica software from these microscopes here, and then the CODEX Multi-Analysis Viewer and VT are also available in the facility. So I'd like to begin with sort of a more basic question about what is multiplexing. And in the most simple of terms, multiplexing is just fluorescent imaging with a high number of probes. So many of you might be familiar with the traditional blue, green, red, far red imaging that is traditional to many microscope systems. These can include things like DAPI, GFP, M-Cherry, or other red fluorescing proteins, and Cy5 as an example of a far red protein, far red marker. And so traditionally, in order to separate signal from each of these emission spectra, you would utilize filters or bandpass filters in order to separate signal from one channel to another. And this is relatively easily done with just 4 probes. Despite DAPI's notoriously long tail, we're able to separate signals from, say, a nuclei from a protein of interest that's been labeled with GFP. This becomes more complex as you try to increase the number of probes in your system to sample more about your system. And you end up getting significantly more spectral overlap between probes. And this becomes more and more challenging to separate each of these channels, and you get crosstalk between systems that makes it more difficult to determine where your true signal is. So multiplexing is a mechanism by which you can more efficiently generate a system in which you have multiple probes, well over 8 probes, that would be able to be spectrally separated, and you would be able to probe your system with a higher number of probes in order to get at more questions that you might have for your research. So this is why you might utilize the CODEX system. First and foremost, it is a multiplexing platform. It can utilize a high number of probes to a theoretical maximum of 84 probes in the system. It's highly flexible. Probes can be tailored to your experiment. It offers whole tissue context within a very large area, up to 20 square millimeters. And this area is imaged at very high resolution. We have robust spatial analysis software available. And as I mentioned, we'll be doing a live Halo demonstration at the end of the talk here. And this is critical for the amount of data that is being generated in one of these experiments. With upwards of 84 probes in a large area, you can imagine that the data sizes of these experiments becomes very, very large, easily in the terabyte range of material, and having a robust analysis software is key for being able to interpret and visualize your data. The system is automated, so in conjunction with the fluid handling system produced by Akoya Biosciences and the Keyence automated microscope, there's no need for user input after you start the run. And depending on the number of probes in the area that you're investigating, this run can take upwards of days. This experiment is repeatable. Tissue is recovered after a run. So if you want to repeat the run, or if you have a downstream application for the tissue after an Akoya CODEX run, you can of course do that. So how is the multiplexing in CODEX achieved? And is actually uniquely achieved with just 3 colors. And it capitalizes on the use of this architecture, by which you covalently label your antibody of interest with a unique barcode sequence, which has a complementary barcode to one of 3 different reporters, either a far red, red, or green reporter. The colors here are actually the excitation colors. So you use blue to excite a green, green to excite a red, and red to excite a far red. So the first step in using CODEX is to actually establish this antibody panel. You can either purchase these antibodies directly from Akoya. They have a short list of antibodies that are commercially available, or if you don't have your conjugated antibody of interest available to purchase directly from them, they offer a kit that you can utilize that allows you to create your own conjugated antibody in the system. But once you've created your antibody panel, and we'll get into more details on how to do that later in the talk, you will then simultaneously label your tissue on a cover slip with all of the antibodies in your panel. So say you're doing something like 20 or more antibodies, 20 more probes in your system, you would put 20 antibodies with the unique oligo barcode onto the tissue. And then in order to reveal them, you would reveal them 3 at a time. So you would add the complementary barcode with the fluorophore to your tissue, which would then reveal those 3 specific probes. You would then image those 3 specific probes. And then you would strip those probes, breaking this annealing bond here between the oligo pairs, removing those fluors, and then you would wash and repeat this process, revealing a further 3 probes, different from the original 3 that you had put on in the first case, and repeat this process of revealing, imaging, removing, and then repeating until you've covered all of the probes within your antibody panel to whatever cycle number you would need. So if you're doing n probes, it's traditionally n divided by 3 number of cycles. The basic workflow for setting up a CODEX run is to first assemble the conjugated antibody panel, then label your tissue using these antibodies, and then perform the automated imaging run. And this is done on our Akoya CODEX automated fluid handling system, as well as the Keyence automated microscope. It's at this point that I'd like to sort of pause and ask, are there any questions so far? Ken, I don't know, are there anything in the chat? I can't quite see the chat.
No, there's nothing in the chat.
Okay.
Wait. There is one. So Brittany Allen Peterson asked, what is the strength of the barcodes versus just having a tagged floor?
So you're able to strip them to remove them more easily. So this bond isn't as strong as, say, having a secondary antibody, because all of these probes are then fixed onto your tissue. And so being able to reveal an individual using an individual 3 with this barcode, it's easier to strip the barcode to remove and then repeat the process so you don't get bleed through between the channels, or between the cycles.
I think she might be asking about the signal strength. And so
I was kind of speaking more to signal strength, as well as I know there's quenching, techniques with light and like hydrogen peroxide that will get rid of the floor so that you can do another round. So I just was curious specifically if that barcode is just ease of taking it on and off. That's kind of the strength there.
I believe it is. I mean, so they use a very light DMSO, I shouldn't say light, they use a DMSO bath to remove the probes from the next cycle. And Akoya has published papers that have shown that they can continuously run these cycles up to, I believe they did 120 cycles at one point, to show that you can still get a efficient labeling with a barcoded sequence even after that many runs.
I think Brittany's talking about this. So there's an approach that you do something very similar. You add 3 antibodies at a time, and then you essentially just bleach the living hell out of the tissue and do it again. And that technology, I don't think, is commercially available, and it hasn't really been used a heck of a lot. And I think largely because it's difficult to get complete bleaching of the probes that you've used. Dr. Bay asked, what is the resolution of the technology? Can it be applied to bacteria?
So we're using a 20x air immersion lens. I don't know if we're to that level. I know that it's not a confocal approach. So we're using a wide field microscope. So you might run into issues with bacteria.
So we're getting subcellular resolution, but it wouldn't be at the level of being able to resolve things within a bacterium. I mean, so for example, you're imaging, at every round you image nuclei, and you get, like I said, you get subcellular resolution, but you're not getting much beyond that with that kind of objective. A third question was, does it work with both frozen and fixed tissue?
It does. So I'll get into that in a minute, but yes, it does work with fresh frozen in OCT or paraffin embedded formalin fixed.
And here's an interesting question. Julie Brothwell asked, when assembling the panel, do you need to be concerned about the order of the antibodies that are imaged? E.g., do you start with lowly expressed proteins and work towards more highly expressed proteins in the different rounds, or does it truly not matter? I can actually speak to that because there's a collaborator that I have in San Francisco that has exactly this problem, where the efficiency with which the probes are stripped can be very important. So if you have a probe that is not stripped between runs, you'll end up mistakenly detecting it in the next cycle. And so, in fact, it is a good idea to look at your dimmest probes first and your strongest probes later, just so that doesn't end up being a problem. And another question is, do you heat the sample to a certain temperature for efficient hybridization?
We do not. The hybrid, so these barcodes are actually designed and validated by Akoya Biosciences. And if you wanted to create your own custom antibody conjugate, you can purchase these pre-validated barcodes from them that have been demonstrated to have efficient annealing and hybridization and dehybridization in this system. So they've done up to 60 antibody or 60 oligo probe pairs, and they've demonstrated that these work well within their system.
So you typically actually buy the antibody kit along with a reporter probe that's going to react with the oligo. So they provide that to you. And I think that's all the questions for now, Connor.
Okay. So at this point, I'd just like to get into some of the details for assembling conjugated antibodies, labeling tissue, and the automated imaging run. So this might address some of the questions that people have, but so the first thing you, of course, want to do when assembling a conjugated antibody panel is to identify antigens of interest. So I'm just listing here a couple of the antibodies that are commercially available from Akoya. including CD3, CD4. A lot of these come from an immune cell background. So that's something that they originally had done to demonstrate the technology. But of course, you can always identify antigens of your interest that you want to include in your panel and create these in a custom manner. As I mentioned earlier, you can either purchase or create these conjugated antibodies yourself. Akoya does have a finite list of pre-conjugated antibodies for both human and mouse tissue. Their conjugation kit is commercially available and capitalizes on this malamide hydrolysis reaction. So in this case, the yellow ball here represents the unique oligo barcode, which you would then apply to your antibody of interest. It's highly recommended that when you are selecting an antibody that you make this as carrier-free as possible to make this reaction as efficient as possible. Ultimately, you take your antibody of interest and you perform a hydrolysis reaction, and you're able to covalently link your antibody to the barcode. And this is how you achieve this, this pair. So once this antibody has been linked to that barcode, you'll have a secondary barcode that's complementary to the unique reporter, which is then assembled into a completed panel where each individual antigen has a corresponding barcode, which you then use in your actual run.
Connor, can I interrupt for a second?
Please.
There's a question as to how tricky is the conjugation of the barcodes to antibodies, and that's really a really loaded question. That was something that, in fact, if you watch their seminars available from the Nolan lab, the people who developed this, and they were saying they had a success rate of something like 50%, which was kind of frightening. But subsequently, Connor's been working with a working group, people all across the country that are working with CODEX, and they've kind of identified a lot of the limiting factors. And the hit rate is much higher than it was. And this working group's been really effective at identifying better strategies, kind of better practices than maybe Akoya would suggest. And so, first of all, they understand a lot more about the conjugation than it's done before. And the most crucial thing was the purity of the antibodies. But something else that Connor might mention to you is that if you end up getting involved with this and you want to participate in that user group, that's a really important, nice resource too. Someone else asked, is the use of cover slips mandatory? So no slides.
Yes, this is true. So I'll get into this in just a moment when I talk about labeling tissue. But yes, we need to image through the cover slip. So we're actually performing the exchange of here. We're actually performing all of this revealing, imaging, and removing on a cover slip inside the microscope. So we do need to image through the bottom of the cover slip in order to image the system. So this does need to be done on a cover slip, and it actually needs to be specially coated. We do use polyol lysine, and then part of the prep for labeling the tissue is a series of fixations, which will all be, it's all available to be trained in this. We offer training for both assembling and conjugating antibodies as well as labeling your tissue, and we can guide you through this. And all of the reagents for each of these conjugation and for labeling are available within the core facility. So there's no need for you to purchase the kit yourself. We have the kit stocked.
And one last question, McKean Reed wanted to know, is there any information about cross-reactivity with rat?
Cross-reactivity with what?
With rat, for rat tissue?
I have not looked into that. I don't know if Akoya publishes anything about their commercially available antibodies for mouse reacting with rat. I can ask someone at Akoya if they have ever done that, but my guess is that they wouldn't.
Yeah.
But sorry, I don't have any information about that.
Yeah, so it's human, it's mouse, and there's also some antibodies that work in paraffin embedded tissues. Is that correct?
That is correct.
Okay,
Yep.
good. That's it for the questions.
Okay. Okay, so we've discussed assembling a conjugated antibody panel. Next is the labeling. And so this is some of the earlier questions. So there are some tissue requirements. They do need to be thin sections between 5 and 10 microns in thickness. As mentioned earlier, they need to be fresh frozen in OCT or formalin fixed paraffin embedded tissue. The tissue must fit on a 20 by 20 millimeter cover slip area. It is a 22 by 22 millimeter cover slip, but we need 2 millimeters on each side for the gasket and the assembly into the actual device that makes it into the microscope. The cover slips are provided by us, and it is rolled into sort of the cost to do a run and do labeling in the facility. As I mentioned earlier, all antibodies in your panel are applied simultaneously. There is fixation before and after the antibodies are added, and general tissue handling for the labeling procedure to get it into an optimal state for your antibodies. And then, as I mentioned earlier, training and reagents are available through the core facility for both conjugation in order to generate your antibody panel, and for labeling in order to put those antibodies onto your tissue of interest. And that's labeling of your tissue. And next, the automated imaging run. So the automated imaging run is handled by two automated systems. First, there is the actual CODEX box, which is a robotic fluid handling system. There is a robotic arm within this box that interacts with a buffer reservoir, as well as the oligo probes that you have preloaded into the system. This arm ends in a robotic needle, and that this needle draws and dispenses fluids in order to continue the flow of the steps here. So the first step is to add the complementary oligofluorophore, 3 of them at a time, to the microscope where your sample sits within this dish. Imaging then occurs on the microscope using the Keyence automated microscope system. This is again automated. Once you predetermine an area of interest, it'll continuously image that at the predetermined point with no need for user input. Once that imaging run is complete, it then strips the complementary oligofluorophore and then washes the tissue, and then repeats this process for each of the individual cycles that you need to fulfill all of your all of your conjugated panel. Here I'm showing you some example data from a mouse pancreas. This field is 7 by 7 or 3.6 millimeters by 3.6 millimeters. And in this experiment, we're looking for immune cell infiltration into islets. As Ken mentioned earlier, DAPI is actually run on each of the cycles in order to do cycle alignment in case there's drift between each cycle. Then in this experiment, we included CD44, TCR beta, Ki67, B220, CD4, CD169, and CD8A. And what I hope for you to appreciate is that we are actually getting significant immune cell infiltration into this islet here. And we can actually stack all 8 of these images together, pseudo-color them into this image. So one of the problems with this is that once you hit about 8 colors, it starts to become difficult for the human eye to distinguish between colors, even though this experiment actually had 14 total probes. Displaying this number of probes becomes difficult as well. But to the question about high resolution, we can zoom in on, say, this islet. And then we can zoom in yet further on, say, this corner of the islet. And what I hope for you to appreciate here is that the nuclear stain in blue has a distinct surrounding of individual signal that is associated with the cytoplasmic space of that individual cell. One particularly challenging part of this tissue is that there's such a high density of the cells within the islet. But we have an array of tools that lets us analyze this tissue. So as I mentioned earlier, we have halo analysis software with a high-plex fluorescent module installed, which helps us visualize all of these markers. As I mentioned earlier, the size of this data becomes very, very large very, very quickly. So if you have 30 probes plus a 20 by 20 millimeter area, you're easily talking in the multiple terabyte region for a size of the data itself. And visualizing the number of markers that you have in one sample can itself be a challenge based on the size and the number of markers. We can then do segmentation and start to identify cells based on the positive or negative probe, based on the positive or negative state of each of the probes within that cell. The Halo software is also equipped with a machine learning algorithm in order to do tissue classification. So you can see here in red that it has identified the islet, green has identified non-islet pancreas tissue, and yellow has identified empty space or just glass. And there's a battery of experimental tools that are within the Halo software for taking advantage of the spatial component of this experiment, where we can do something like an infiltration analysis. So once the machine learning has classified the exterior of the islet, we can then just say, well, how far into the islet is various cell types making it? Are they making it into, what's the distance in which they're making it in? And we can do calculations based on that. We can also visualize a heat map of, say, immune cells within the islet. Or we can do cell proximity analysis, where we ask the distance between two different cell types. It's at this point that I'd like to jump into a live Halo demonstration. But before I do, I just wanted to highlight the staff here at the ICBM. If you have any questions, please feel free to contact us. Please feel free to visit our website for more information about the systems in our facility. And this is our general helpline if you wanted to ask us about any of the other microscopes or CODEX in particular. Is there any questions that I can address at this time before I go to the Halo demonstration?
Let's see, did you say this can only be done with a 20x objective?
Yes. It is
Is that
limited
true?
to,
We can't change objectives on the system?
it's baked into the software, unfortunately. It's something, it's a limitation that is linked to the software to process the data.
Okay. Yeah, this is one of the problems with dealing with Akoya is that everything they do, it's just a robotic fluid handling device. You think you could hang it off any microscope, but they've seriously embedded everything, all the fluid handling with the microscope control. So it's pretty non-negotiable what kinds of things they'll let you do. That's the, oh, wait, what is the standard process for a PI to analyze and store their large data files?
That's an excellent question. So we've actually been working with IT to generate temporary data storage facilities that would be able to handle large data. We can set up geode test spaces that are exactly suitable for storing this data temporarily. But we also have data pipelines that are available for people if they just want to use a SharePoint or a Teams SharePoint for their lab. If you have a specific request, I'm kind of been the point person on interacting with IT and some of the data storage solutions that we have available. So if you have some specific question, I'm more than happy to address that.
Jill asked, can you comment on optimization steps that are needed for the antibodies?
Absolutely. So generally speaking, there's a suggested starting point for the exposure times for each of your probes. And Akoya does recommend that you spend time optimizing. So you would run one cycle or run one entire automated system run. But then once the tissue is recovered at the end of the run, you can then look at the data and then make adjustments based on the exposures that you see in the first run, and then further optimize the second run.
And Jeffrey Bloodworth wants to know, does the software determine proximity to a predetermined quote unquote other cell, or can it perform unbiased nearest neighbor analysis?
I'm not sure about unbiased nearest neighbor, but you can determine, you can assign cell identities within Halo and then do like distance between two cell types. I'll show you in just a moment what exactly I'm talking about when it comes to the distance between two cell types.
Li Zhang would like to know, in your experience, does a set of antibodies work in different organs? Antibodies for spleen work well with pancreata, for example.
I can't necessarily speak to your particular system, but I can say that people, if you have a preferred antibody that you want to utilize within the system, or one that you know works in an immunofluorescence model, we can actually adapt that antibody to the CODEX system. There are commercially available kits that can clean up existing antibodies and remove carriers, that if you had a favorite antibody that you wanted to see conjugated and included, we can do that.
And also, I think, Connor, you've looked at pancreas, you've looked at a couple of other organs, you looked at kidney, and if I understand correctly, most, if not all of the antibodies that have worked in one organ that we've acquired from Akoya have worked in the different organ systems as well.
Yeah, I haven't seen one that has specifically failed, but because of an organ difference, I think that it's going to be specific to the quality of the antibody, really.
Hangzhu asked if, how harsh is the condition to strip the reporter? After a couple of runs, will the tissue be damaged?
So as I mentioned earlier, Akoya has published, well, I should say the Nolan lab has published several reports that suggest that after iterative runs in the over 100 runs that the tissue isn't damaged, it is pretty aggressively fixed during the labeling protocol. The conditions to strip the oligo is 80% DMSO and 20% about PBS. So it is a pretty harsh stripping, but we have not seen tissue degradation as a result of the stripping at all.
So actually, that's one of the things that if a person is really interested in this, they should look at this as paper that came from the Nolan lab, the Christian Church's paper. Maybe we can email.
Yeah, I mean, yeah. If you want to email me, I'd be happy to forward that paper to you.
They characterized the hell out of this system. I mean, they did hundreds of runs and they characterized the fluorescence level over time, and they did a very, very rigorous job. The tissue is incredibly stable. They did comparisons of when they probe per probe in the first run versus the 100th run, and they got equivalent levels of labeling. So it's pretty impressive. Brittany Allen and Peterson wants to know, if you stain H&E at the end, will the system be able to scan that as well?
I'm sorry, I kind of broke up there. Can you say again?
If you stain for H&E at the end, will the system be able to scan that as well?
So unfortunately, you cannot do H&E staining before you do a CODEX run, but you could do it after a CODEX run because the tissue is recovered.
And there's no reason we couldn't collect an image of that you could then align with the CODEX.
That is true. Yep.
Okay. How long does it take to run one slide, say with 21 probes? The size of the tissue.
It varies greatly. So one cycle is about an hour, depending on how much area you are running. So if you wanted to do 21 probes, that's roughly 7 hours. But again, it's an automated system. So you could set it up in the evening and then come and pick up your data in the morning of the next day.
And another question, hybridization of the reporter and barcode has to be at a certain temperature, otherwise it would reduce the specificity among the 3 probes. I guess that's more of a comment than a question.
Well, so I guess to speak to it, according to Akoya, these are optimized for room temperature, annealing under the conditions of their buffer system. So in their system, they're using a proprietary buffer called CODEX buffer, which has been specially developed and designed to facilitate annealing at room temperature to show that they're actually, and they do demonstrate this in that Schwartz paper, which I'm happy to send out if anyone is interested. But they do, and we've done internal benchmarking where you do see that the probes do align with the expected location of the probe, and then they are efficiently stripped when we run a blank after that run.
Con has cut to the chase and asked, how much does it cost? I assume that's something you're going to talk about at some point.
So I wasn't going to touch on cost initially here, but I can tell you that it'll depend on the number of antibodies that you want to run. If you want to send me the panel that you're interested in running, I can do a cost estimate for each of you individually if you want. I'm happy to do that. I can break down how much it would be per antibody, per labeled tissue, and per run.
But can you just say, in terms of if someone's doing 20 probes and they're using Akoya antibodies, just ballpark, some kind of idea?
Sure. So the antibodies that are from Akoya are around $400 per pre-conjugated antibody with the oligo probe combo. So you would get both for about $300 or $400 depending on the probe, and then it's about the same cost, including an antibody cost, to generate your own. It might be a little bit more expensive depending on what quality antibody you decide to go with. And then for an actual labeling run, it's about $350 per tissue, and an actual run, it varies greatly. We charge by the number of cycles, so that'll be dependent on the number of antibodies you have in your panel. But it's about $100 per cycle.
So the major cost is the cost of the reagents. It's the antibody cost.
Yes. Yeah.
That's the thing that drives the costs more than anything else.
Right. But I will remind people that once you have your antibody panel established, that's good for about 50 runs. So if you have your antibody panel made, you're more than set up to do any number of N and tissues that would then propagate that cost.
So if I can interrupt one more time here,
Yeah.
This is what makes it difficult to get people to understand how much it would cost to actually try it. It's not set up very well to try it, because basically there's a certain amount of antibodies you need to buy. And at that point, you're set to do studies for a long time. So if you just want to try it once, we actually have some antibodies I think we can probably work with you on. But the point is that this is the kind of thing that groups of people would do well to work together and share reagents, unless they really plan on doing something. Because when you buy one of these pots of antibodies for $300 or $400, you've got a lot of CODEX runs available to you. So it's tricky to work it out, and I would suggest that, I don't know if this is something that Connor's going to talk about or not, but this is something that I think people should be thinking about using CTSI funds. There's a core use grant deadline coming up April 1st. And so people that are interested in applying this technology should be in touch with me, because I have to write a letter of support. There's something up to $10,000 per PI that's available to support use of core facilities, which could include using the CODEX in our facility. And people can be in touch with me if they want to figure out how to do that.
Yep, and that would cover costs associated with training or reagents to do the conjugation or labeling, or the actual run itself.
Okay, and then Ed Greenfield said, when you refer to a run, does that mean one tissue section?
Yes, so a run is all the cycles that you would need to do one tissue, yes.
Another question, can't we just mount the section on positive charge slides, or you require a specific slide for the mounting? We provide the cover slip for that.
We do provide the cover slip. We have not specifically tried positively charged slides, but it is recommended that you use poly-L-lysine from Akoya Biosciences.
Okay, that's where we are now in the questions.
Okay, so let me try and transition here to the CODEX. Here we go. All right, can you guys see the screen here?
Yeah.
All right, great. All right, so this is that same tissue that I was showing you before. This is the mouse pancreas tissue with an islet. Right now I have all of the channels selected, and you can see that it's kind of messy. So I'm actually going to manage the channels using these check marks here. And you can also manage the channels colors here, as well as their relative intensity to each other. I'm actually going to show and hide all the channels in order to turn them off. So here you'll just see them briefly one by one tick down, and you can see each channel fade out. And then I'm just going to re-enable DAPI, and then zoom in to show you sort of the resolution that we have here. And then we can turn on individual probes, like say CD45. And then the way you can do segmentation is doing what's called a real-time tuning, which is in this analysis window here. So first I'm going to select CD45. Of course, I can't find it. There it is. And then make sure that I'm visualizing on CD45 here, and then doing real-time tuning. So here's a real-time segmentation that is occurring. So each white space is actually the proposed distance from the segmented nuclei. This is all tunable, so you can change how aggressively you're segmenting your nuclei. And then you can also tune your threshold for positive within each of your sample, each of the probes.
How is the border of the islet defined?
Sorry, I can back that out. That's actually using the machine learning algorithm. So this was a machine learning algorithm that was trained on this tissue to identify the islet tissue, the non-islet pancreate tissue, and the glass.
And Connor, I assume that you end up getting counts and characteristics of the segmented cells?
Yes, yes. So I'm going to go to, so once, well, so I'm kind of jumping the gun here, but once you've gone through each of the probes and you've done thresholding for each one, you can run that analysis and you get this, where you get a total number of cells. Total cells here is 44,000 within this tissue, but then each of the cells are segmented and classified based on their positive or negative probe value. And then you can turn off the filters in each one of these. But I just wanted to make sure that we got the real-time tuning in here. So for this, you set up a threshold. So every time you start at a threshold that allows none of it as considered to be positive. So this is what you would start with. You would first set up your nuclear segmentation. And then you can start to classify by saying, I'm going to set a threshold, say here. And then it'll identify anything in yellow has been considered positive for CD45. Let's test here where we see some positive and negative samples. Let's see, so we can then validate this and say, okay, I think that I'm getting good segmentation and a good labeling here. I think that one's positive, so I'm going to try and decrease the threshold to include that cell. And computer processes. There, that looks better to me. So this patch of cells here looks negative to me, and it's not showing up as positive. So I'd say this is a pretty good segmentation and thresholding. And you can do this for each of your cells, or each of your probes, and then run the analysis over the whole tissue, and you get something like this. And so right now I'm displaying sort of individual cell identities. Let's see. So one of the things that I showed in the presentation was this heat map. And here we're actually showing a heat map for CD8A, CD44 positive cells. And then the infiltration analysis is shown here. And then you get quantitative data from this. So in this instance, any negative value is actually the distance internal to the islet. So the first bar is this first green, and then as it rainbows inward, you get more and more. This can be extended inward to cover the entire islet. But you can see that there are some of these double positive cells on the outside, and that they are concentrated to the interior of the islet. And then you get all of this in a table as well. So you can export any number of these data tables out of the software.
So Connor, most of the antibodies you're showing here, I believe, are antibodies you can buy directly from.
They are. These are all commercially available. And
these are a lot of them are inclined towards immune cell localization. But
correct.
We've been working with the Diabetes Center for a while, and I believe haven't they developed some antibodies, or at least they're working on developing antibodies to identify beta cells, alpha cells, and that kind of thing as well. Right?
Yes, indeed. So I mean, really, any antibody that works for you in another context can be applied to this system.
Okay, I have a question. Is HALO included in those costs we talked about earlier? Is HALO something the core analyzes or user? If we need to reanalyze something, will we have continued access to that software? So how does the analysis work? So
HALO is just like any of the other software packages in our facility. They're built into one of your workstation computers, are charged at a $25 an hour rate. We are also available if you want to hire any of the core faculty, core staff. You are welcome to do so, and we would be happy to guide you through any of this process.
Ed Greenfield asks, I'm sorry, to compare multiple sections, are the segmentation thresholds comparable?
To compare multiple sections. So
in other words, I think what he's asking is that if you wanted to do, if you want to compare multiple samples, and are you going to be able to use a single segmentation set of parameters across all the different tissues so you can compare the results?
So yes, so you can save the segmentation parameters as well as the thresholding values, and then apply them to any other tissue that you run.
So practically you can, but in answer to the question, Ed, I think it's going to come down to how consistent your tissue is. because at the end of the day, the segmentation that's worked for one particular sample may not be the same due to things like autofluorescence and the quality of the tissues. So that's something I think you're gonna have to do on a case-by-case basis.
Right, and you can always adjust and reanalyze. So if you have a positive control or something like that that you can benchmark against, and then have that as your master analysis settings, and then apply that to your experimental tissues, that would be great. But if you don't have that kind of condition, you can always reanalyze under different conditions each of your tissues.
So that's the kind of stuff I was getting at, but what I was really thinking about is the labeling and releasing and relabeling and all that stuff reproducible enough so that you could compare different sections using the same threshold.
It would depend on your tissue. I mean, it's, you know, in our hands, the tissues that we looked at that would be considered comparable, we're getting similar levels of labeling. But at the end of the day, the labeling itself is very robust and consistent. But the thing that's going to vary is the tissue quality.
Yeah, it'd be the same kinds of tissue, but maybe from, you know, an experimental mouse versus control mouse.
Mm-hmm. Yeah, well, to the degree that the tissue is as fresh, it's been fixed similarly, and then there's going to be issues like, is there more autofluorescence in one tissue than another tissue? But to the degree those things are more or less consistent, then the same thresholds can be used across that. I mean, it's something that we're encountering in some studies that we're doing with the kidney, where we're looking at some injured human kidney biopsies that have enormous amounts of autofluorescence, which can be, and it varies enormously between the tissues. So a threshold that's worked to identify, like say CD45 in one tissue may or may not apply to another tissue because of this obstructive autofluorescence, which is something you have to cope with. In the best of all cases, you're right. What you really would like to use is the same segmentation parameters across all your tissues, the same thresholds for all your tissues. That makes it the most comparable experiment. But at the end of the day, there are just occasions where the biological variability in things can influence the results. That's something you just have to deal with, like any fluorescence experiment.
I can also add that you can label both tissues simultaneously. So you can do, you know, any number of tissues simultaneously in the labeling process. So they're as handled by the same hands, same reagents on the same day, to try and increase that consistency.
Could there be image normalization used to compare across different tissue treatments and tissues? I don't. Anne, you want to explain what you mean by that?
Yeah, so that's often what we do, right? When we have different sets of data, that we have something that we normalize our data to, that we can compare it across tissues, so that maybe they can't be direct measurements, but they can be normalized measurements. So maybe there's some threshold of some marker that you use as your standard, and then everything else is based on a relative difference between that level.
Yeah, I mean, I think there's nothing inherently different about CODEX fluorescence imaging than immunofluorescence imaging. And in fact, I would argue that it might be more linear than immunofluorescence imaging. So I think whatever techniques that you've used to get normalization across different tissues, you should be able to apply here as easily as for anything else. Like, there's nothing special to CODEX imaging in that sense.
I'll also just add that each of the data tables that are, so on an example here where you've segmented and thresholded each of your probes, you can then get one massive table here where it lists each of the intensities relative in relative units, as well as the percent of cells that are positive, and you can also gate on here. So for example, if you wanted to identify all of the CD11B positive cells, you can select those, and then I'll just highlight all of them, and you can see that these are all the CD11B positive cells, and then you can add further gating from this point. If you click an individual cell, it'll highlight that one, but you can then say, I want all the CD11B and CD44 positive cells. Now we've added that. Now you can see that these are the positive cells that are these are the double positive cells.
So with respect to Anne's question, I guess the other thing to consider here is that there's an enormous amount of capability in the Halo software, but to the degree there's something that you need to do with the data, you can output this data in the CSV file, I assume, right?
Yeah, I mean, it can be exported directly into Excel.
Yeah, so the point is, to the degree you need to do some kind of manipulations of the data, the raw, the measurements themselves, all the data is exported, and you can do that as you would with anything else.
Are there any other questions?
Not right now.
Okay. Well, that's kind of where I was going to wrap up. I'll put this back up on the screen here. If you have any additional questions or would like assistance in generating a panel or want to engage with training or experimental design, please let us know. We'd be happy to address those questions.
Wait a minute, we
one
have
more.
some panic questions at the end here. Without machine learning feature, how would you distinguish any areas?
So you can actually manually annotate the system as well. You don't have to necessarily use the machine learning algorithm, but you can manually annotate as well.
And someone was asking, I only see Psi5 reporters that is available in a coil biosensors. Do you know, are there available with any Psi7 reporters? Our system isn't set up for imaging Psi7, and the Psi7 I actually have set up mostly for paraffin-embedded tissues. Is that right?
That is correct, because when you have paraffin-embedded tissues, the autofluorescence of the paraffin interferes with that peak, and so you would shift to a Psi7. But we don't have a Psi7 filter on our Keyence. Instead, what we do is we work around it using other fluors that are available. And I'd be happy to help you if you had a paraffin-embedded tissue to design the panel.
Ed Greenfield asked, how good is the quantification regarding probe intensity single cell resolution? So you get single cell resolution. The segmentation is based on a Voronoi tessellation around the nuclear images. So the regions of interest are determined by that tessellation. The sensitivity versus other platforms, protein of RNA, I haven't actually seen a comparison. The linearity, you're collecting the images on, is this a CMOS or a CCD, Connor, do you know?
It's a CCD, I believe.
Yeah, and to the degree you haven't saturated anything, the signals are linear. At least they're linear to the degree that the antibody recognition of the epitope is linear. And so essentially it's going to be equivalent to what you can expect from immunofluorescence. And so whatever caveats there are around the amount of fluorescence versus the amount of epitope, it still pertains. So I think, nope, better just keep on being more questions. Do you see degrading signals after multiple probe stripping and reprobing? No, and we haven't really characterized that very carefully, but certainly again, this paper by Church was astonishing, where they did many rounds and then compared the signal they got when they probed for something in the first round versus the last round, and they got very similar amounts of signal. So the stripping and reprobing doesn't seem to affect signal strength. And I think that might be it, Connor. And anybody that has any specific questions should contact us. I encourage you, particularly if you're really interested in this, in how the sausage is made, that you look at the Church paper, which is incredibly complete. In fact, it's more complete than a COE would prefer it to be, I think. And get in touch with us. We can help explain and help you work through this. And again, I want to reemphasize the idea that one of the ways that you can get into this and try it out would be through the CTSI funds that are available. But the window of time is closing in on that because, my God, it's due next Thursday, which is to say that if you need a letter from me, I'm not going to want to hear from you much beyond the first part of the week. So this needs to happen quickly. All right. Great talk. Thanks. Terrific. Good. Someone complimented your talk, Connor. Thank you very much, Connor. This is terrific. I appreciate it. And also thank you, Jill. I appreciate the opportunity to move this in front of people.
Thank you all for your questions and thanks for attending.
All right.
Right.
Thank you, everyone.
Bye-bye.
Take care.